Hi everyone, welcome back to my channel, and today we are going to learn about ketone bodies. In this video we will discuss about how ketone bodies are synthesized, what is this ketone bodies, how is ketogenolysis occurring, and in which conditions these ketone bodies are increased in our body. So we all know that glucose and fatty acids, they serve as the fuel energy for our body. But apart from these two components, there is ketone acid, which also serves as the fuel energy for our body. There are three kinds of ketone bodies. One is acetone, another is acetoacetate, and the third one is beta hydroxybutyrate. One very important thing that we have to know is these two ketone bodies, acetoacetate and beta hydroxybutyrate, serve as fuel energy. But this acetone does not serve as the fuel energy. Rather, it is very important in diagnosis of making ketoacidosis because it is excreted through our lungs and it gives fruity acetone smell. So in case of ketoacidosis, a patient can have fruity acetone smell. So it's important for making diagnosis, but it is not important. Does not serve as the fuel energy for our body. These ketone bodies are water soluble and they yield the energy. Now let's learn how they are synthesized. These ketone bodies are synthesized in liver. So these ketone bodies are synthesized in liver, and whenever there occur beta oxidation of fatty acids, then it produces acetyl-CoA. 2-acetyl-CoA by the enzyme beta ketothiolase will make acetoacetyl-CoA. Now this acetoacetyl-CoA will convert into HMG-CoA through the enzyme HMG-CoA synthase. Now this HMG-CoA will produces acetoacetate, one component of this ketone bodies, through the enzyme HMG-CoA lyase. Now there will be spontaneous formation of acetone from this acetoacetate, and through the enzyme acetoacetate dehydrogenase it will form beta hydroxybutyrate. So this is how this ketone body synthesis occur, and it occurs in liver. And we have already discussed about this acetone that does not serve as the fuel energy. Rather, it is important in making the diagnosis of ketoacidosis. Now, we have already discussed in our previous videos that fatty acids is not utilized in RBC and brain. But in case of ketone bodies, this RBC can't utilize the ketone bodies because during this synthesis of ketone bodies in liver, they have a lot of enzymes, and these enzymes are produced in mitochondria. But we all know that RBC don't have mitochondria, so they can't utilize these ketone bodies. But this brain can utilize ketone bodies, but it can't utilize the fatty acid. So in another word, we can also say that fatty acids, through becoming ketone, serves as the fuel energy for brain, but directly it can't use as a fuel energy for brain. Normally in our urine, no ketones are detectable, but in certain conditions it is detectable in case of diabetes, ketoacidosis, starvation, alcoholism, or in case of diabetes where this uncontrolled diabetes mellitus condition. Now, individually we will discuss how this is occurring. So this is the small part of TCA cycle. This malate is converted into acetoacetate, and acetoacetate through this combining with acetyl-CoA will make citrate, and this citrate will make isocitrate. Now very important thing is this one step is a reversible kind of step. This oxaloacetate can convert it into malate whenever there is high level of NADH. This is very important thing to know. Okay, let's discuss. In diabetes, there will be low insulin, and whenever there will be low insulin, this acetoacetate will go in formation of glucose via the process of gluconeogenesis. And in case of starvation also, what happens is there occur the beta oxidation of fatty acids, which will produce a lot of NADH, which reverses the formation of acetoacetate to malate. It reverses process. Normally what happens is malate is converted into oxaloacetate, but whenever there will be high amount of NADH in case of beta oxidation of fatty acid, it reverses this process and this oxaloacetate will go and make malate. So what happens in this case is this oxaloacetate is depleted. Now whenever there won't be any oxaloacetate, this acetyl CoA uptake is decreased in this TCA cycle. So this TCA cycle process will slow down. So the excess amount of acetyl CoA forming will go in the process of this ketone body synthesis. So more amount of ketone bodies will be synthesized. Likewise, in case of alcoholism, in the process of its metabolism, a lot of NADH are produced. So the same thing happens and it will result in formation of the ketone bodies. Now let's know about some terms. What is ketonemia? When the rate of synthesis of ketone bodies exceeds the rate of utilization, its level increases in blood, and that is the term ketonemia. Ketonuria means whenever ketones are excreted through the urines and is detectable in urine, that is the ketonuria. These both terms means ketosis. Let's learn about ketoacidosis. This acetoacetate and beta hydroxybutyrate, these both are the strong acids and they will lower the pH of our blood. So ketoacidosis happens, and usually due to starvation it won't produce any ketoacidosis usually. And in case of ketoacidosis, rapid treatment, rapid correction is very important. Now let's learn about ketogenolysis. We have already discussed how ketone bodies are synthesized. This acetoacetate through the enzyme acetoacetate dehydrogenase will produce beta hydroxybutyrate. So now this beta hydroxybutyrate will oxidize to acetoacetate, and this acetoacetate will convert into acetoacetyl CoA that will result in formation of 2-acetyl CoA, and that will go in the formation of in the process of TCA cycle. And another important thing that we have to know is synthesis of ketone bodies occur in liver, but ketogenolysis, it occurs in extrahepatic tissues. Because for this oxidation process, when beta hydroxybutyrate is converted into acetoacetate, this enzyme is not produced in liver, so the ketone bodies can't metabolize in liver, so they are metabolized in extrahepatic tissues. So this is all for today. I hope you guys enjoyed, and we will meet up in next video.